Connective tissues are of mesodermal in origin and develop from the mesenchyme. All tissues of adult body derived from the mesoderm. The principal function of connective tissue is to bind other tissues together and to give support to various structures in the body. 20% part of the body is formed by the connective tissue. Connective tissues are distinguished by the presence of matrix or intracellular material. Here in photograph you can see a small mesenchyme cells from which they derived a different loose connective fibers. These are the fibroblasts. These mesenchyme cells turn into a fibroblast and they produce fibers, and now they term as a loose connective tissue. The connective tissues of cells themselves do not form the greater part of tissue. It is the intracellular substance produced by the cell that furnishes the main bulk of connective tissue mass. The intracellular substance consists of fibers and a large amount of an amorphous, transparent, gelatinous ground substance. It is made up of acidic mucopolysaccharide bound with proteins to form hyaluronic acid. The tissue fluid in connective tissue remains trapped between the molecules of hyaluronic acid. Several types of cells, some wandering in a matrix, other fixed, are found. 3 types of fibers may be present in matrix. Here, cell lineage from mesenchymal cell is shown in this photograph. You can see a mesenchymal cell, a parent cell from which different macrophages are developed. Some are fixed and some are wandering. These are macrophages, means WBCs. Fibroblasts are developed, which forms the loose connective tissues. Adipocytes are developed, which form the fat storing cells. Chondroblasts are developed, which forms bones, osteoblast, and endothelial cells, which forms the blood vessels. Mesothelium cells, mast cells, all these connective tissues are developed from the mesenchymal cells. They also develop this hemocytoblast. From hemocytoblast, thereafter develop different blood cells like neutrophil, eosinophil, basophil, erythrocytes, plasma cells, lymphocyte, monocyte, megakaryocytes. Basically, origin of connective tissue is a mesenchymal cells which forms intermediate blast cells which then convert into a bones, fat cells, fibers, blood cells, WBCs. There are 3 types of connective tissues: loose connective tissue, dense irregular connective tissues, and dense regular connective tissue. Now here this name terminology is based on arrangement of connective tissue fibers in the tissue. You can see here they have shown a different arrangement of fibers and a matrix in a square cube. If it is these fibers are loosely arranged, it is a loose connective tissue with lot of matrix, less fibers and more matrix. Here densely, compactly, irregularly arranged connective tissues fibers and within the matrix, and here these fibers are very perfectly arranged, so regularly they are placed, so called as a dense regular connective tissues. The example is given here. Very dense regular connective tissue example is tendons, ligaments, cornea. In case of dense irregular connective tissue, example is a dermis of skin. And loose connective tissue, example you can see in mesentery and omentum. Mesentery are a thin membranous structure present between the two organs in our abdominal and thoracic cavity. This is a actual color photograph of connective tissue where we can verify the arrangement of these fibers in a matrix. Here they are very loosely arranged. The dark spot indicate the nucleus, and here they are very compact and regularly arranged, and here they are arranged in systemic way in a parallel manner. Dense irregular connective tissue, dermis of skin. Dense fibrous connective tissue. In this dense fibrous connective tissue, the amount of matrix is very much reduced and mostly fibroblast cells are found. Due to abundance of fibers, these tissues possess a great tensile strength. On the nature of fibers, this tissue is of two types. White fibrous connective tissue. In the matrix of this tissue, only collagen fibers are found. This tissue provides strength and found in tendons, which connect bones to the muscles. In higher mammals, while fibrous connective tissue occurs in a dermis from which leather is prepared after tanning. The periosteum of bone and the perichondrium of cartilage are made up of this tissue. It is also occurred in fibrous capsules of penis, testes, eyeballs, blood vessels, and glands. Another type is a yellow elastic fibrous connective tissue. In the matrix of this tissue, yellow elastin fibers are found. These occurs in highly elastic and mobile structure like ligaments. Here in this section of elastic connective tissue in blood vessel wall, you can see a presence of internal elastic lamina. This characteristics of internal elastic lamina is it expands when required. It maintains the elasticity and strength of blood vessels. Connective tissue fibers. Now all these connective tissue are made up of very different types of fibers. In this, majorly a collagenous fibers which are in a bundle form, reticular fibers which are in network form, and elastic fibers are there. Elastic fibers, they are anastomosing in bundle form. In color photograph of a section, you can see a regularly arranged collagenous fibers. Here you can see the regularly arranged collagenous fiber. Here you can see a network of fibers which are interconnected and a very precisely structured elastic fibers. Reticular connective tissues. Now here in this section in photograph we can see presence of this reticular fiber. They intervene in the tissue and they form networks to provide support to the tissue. Collagens, these fibers, they form bundles. Inside there is a fiber, that small part is a fibril and the smallest one is a tropocollagens, which form periodic bundles and they together form microfibrils. Under micro photograph they look like this. Because of these striations we can identify the collagen fibers. In loosely arranged collagen fibers you can see a presence of a fibrous structure. Under scanning electron micrograph of collagen fibers bundles you can see a presence of fibers here. A fibroblast. Now all these fibers are synthesized by the fibroblast. Fibroblast original cell is mesenchymal cell. From that it develops fibroblast, and this fibroblast then synthesize these collagen fibers. So in this earlier slide, whatever this fiber you see here, they are synthesized or produced by this fibroblast. A collagen type, different types of collagens are there. In this electromicrograph again you can see the presence of a fibroblast collagen fibers you can see here. The fixed connective tissue cell types. A fibroblast, fiber producing cells, fibrocytes which mesenchymal cells, pluripotent which fixed macrophage histiocytes and adipocyte fat storage cells. All these are the development of mesenchymal cells to form the different connective tissues. Now fixed connective tissues cells, in a section you see this hollow space. This hollow space is actually a adipocytes, which is a vacuole like structures which stores fat, and so called as a adipocytes. And supporting structures they have shown here is a fibroblast, fibers and a nucleus, that is fibroblast nucleus and the fibers which support these adipocytes. Then macrophages, under light microscope you can see macrophages like this, and under electron micrograph you can see macrophages like this. Here you can notice the presence of macrophages, that is a connective tissue. Mononuclear phagocytic system. Mononuclear phagocytic system, reticuloendothelial system, where mononuclear wandering cells, they form these giant cells, and macrophages which gives spleen, lymph node, bone marrows, connective tissue, lung, serous cavities, osteoclasts that form bone resorptions, Kupffer cells, liver sinusoid cells, central nervous system, sinusoidal endothelium, liver, spleen, marrow, all characterized by the phagocytic activity. They works like a WBCs. Lymphocytes and plasma cells. They are agents of immune response. They all related with the immunity of our body. So this lymphocyte differentiation stimulated by immune activations. Lymphocytes form the plasma cells that produce immunoglobulin antibodies. Under microscope they look like this. Here it is a lymphocyte which when stimulated it forms these plasma cells. Eosinophils, again a connective tissue, actively phagocytic, destroy immune complexes, antigen antibody. Mast cells, the secretion products, slow reaction substance of anaphylaxis, heparin, anticoagulant, histamine, stimulate inflammation, serotonin, vasoconstrictor, promotactic factors are produced by the mast cells, which is a connective tissue. So in this slide we see different types of connective tissue cells. Here is a plasma cells, here is a lymphocytes, is a macrophage, this is a electromicrograph of eosinophil, this is again micrograph of mast cell. Adipocytes, adipose or fatty connective tissue, it is really a loose connective tissue in which fat cells are particularly abundant. They take the place of other connective tissue cells which are therefore far less numerous than otherwise. Adipose tissues help in storage of natural fat, which is then available as a source of heat and other forms of energy for the body. Adipose tissues forms about 50% part of body. In whale and elephant, beneath skin at forms bladder. In man, near the kidney, around the kidney, there is a fat bodies. In sheep, it forms thick tail, and in camel, it forms a hump. There are two types of adipose tissue, white or yellow fat. It contains large fat cells, each having a large and single fat globules. Brown fat, it contains fat cells, each having several small fat globules. It is present in such region as the neck, between the scapulae. Pigment incorporated in fat is responsible for color. Some investigators have referred to the brown fat as a hibernating gland. The color is due to presence of cytochrome, which is an iron compound. Brown fat produces 20% energy than the white fat. Blood is also one of a connective tissue. It is called as a liquid connective tissue or vascular tissue. The vascular tissue comprises blood and lymph. Though the cellular elements are free and the intracellular substance is fluid, that is plasma, both cells and fluid course through the body in vessels which are lined with endothelium. The liquid connective tissue differs from areolar tissue as the matrix is liquid and called plasma. Fibers are absent in matrix or plasma, and the cells found in plasma are called corpuscle and float in the plasma. The corpuscle do not secrete plasma. Blood consists of a fluid intracellular substance, the plasma, 54%, in which are suspended the blood corpuscle around 46%. Approximately 7% of normal body weight is accounted for the blood. Plasma, the fluid portion, that is plasma, comprises about 91 to 92% of water and 8 to 9% solid. The solid consists of 7% protein, 0.9% inorganic salts and miscellaneous organic compounds. It is due to inorganic salt. The pH of blood is 7.4, that is alkaline. Plasma proteins, globulin, albumin, fibrinogens and prothrombin occurs in colloid form. These proteins are formed in liver. Salts are found as sodium chloride, bicarbonate, sulfate, phosphate of calcium, potassium, magnesium, and some miscellaneous substances are like nutrient substance like glucose, vitamin, fat, amino acids. Besides, certain excretory products like urea, ammonia, uric acid also found. Enzymes, hormone, gases are present in plasma. Blood corpuscle or hemocrit. These form 46% of blood. The corpuscle can be divided into four types: erythrocytes or red blood corpuscles. Erythrocyte, the shape and size and number of RBC varies from species to species. These are found mainly in chordate, although in some invertebrate too you see this. In mammals, the erythrocytes are non-nucleated, biconcave and smaller than that of other vertebrate. The smallest RBCs in animal kingdom is that of musk deer, measuring about 2.5 micron. In camel, the RBCs are nucleated and oval in appearance. In rabbit, these measures 6.9 micron, in man 7.5 micron. During the process of their formation, the nucleus, mitochondria, ribosomes, centrosomes, etc. all are lost, leaving behind stroma capsule. Since the RBCs are non-nucleated, it is sometimes said that they should not be called cell. The name erythroplastid may be used, but erythrocytes is the more common term. The total number of erythrocytes in an average human being is estimated 30 trillion. Each RBC is bounded by a plasma membrane. Internally, it has a framework of protein, stromatin, lipid and cholesterol. About 26 to 27 crore molecules of hemoglobins are packed in this framework. Thus, about 34% of weight is formed by hemoglobin and water forms about 60% of corpuscle. The hemoglobin is made up of heme and a protein globin. Heme parts is derived from the porphyrin and iron, which is ferrous. The hemoglobin contains 4 polypeptide chain, alpha containing 141 amino acids, beta, gamma and delta chains, each having 146 amino acids. The molecular weight is 68,000. The hemoglobin is a respiratory pigment and 1 molecule of hemoglobin carries 4 molecules of oxygen. The lifespan of RBC is 100 to 120 days in mammals. Spindle cells or thrombocytes. Blood contains thrombocytes which play an important role in blood clotting. These are of approximately the same size as small lymphocytes. The cytoplasm is clear and the nucleus is oval or rounded and take dark staining. The nucleolus is absent. Agranular leukocytes. The cytoplasm of these corpuscles is clear, without granules, and the nucleus is roughly round or indented. These are of two types. Lymphocyte, these are small, roughly rounded corpuscles comprising about 20 to 25% of total leukocytes counting. These are less motile. The nucleus is spherical or rounded, surrounded by a thin layer of cytoplasm. These cells have a lifespan of 1 to 3 days and produce antibodies. Another one is a monocyte. Here, they appear to be a somewhat like large lymphocyte but have more abundant cytoplasm. A nucleus is comparatively small in size. The nucleus, which does not stain very deeply, is sometimes markedly constricted. They form 3 to 8% of leukocyte counting. These are actively motile and help in phagocytosis. Owing to their amoeboid facility in assuming a variety of shapes, they are able to squeeze between even the irregular edges to edges margin of flat endothelial cells, forming the thin wall of capillaries, thus escaping entirely from closed blood system into the extracellular fluid or lymph everywhere in their phagocytic forays. This is called as a diapedesis. Granular leukocytes. In the cytoplasm of WBC, granules are present. These are of three main types, although integrating forms are abundant. They are rather similar in size and are distinguished chiefly by the staining properties of granules which they possess. They are of different types. Neutrophils, these are often called as a heterophils in the human beings. They form 60 to 75% of total white blood count. Their lifespan is for 2 to 4 days. Their granules are very fine and stained with a neutral dyes. The nucleus shows great variations in shape but usually appear in the form of three lobe-like regions connected with delicate strands. Term polymorphonuclear leukocyte is frequently applied to this type of white corpuscles. The neutrophil help in phagocytosis of bacteria. Eosinophils. The eosinophils forms 2 to 4% of total white blood counting. Their lifespan is for about 8 to 12 days. The eosinophils are found in all the vertebrate except certain species of fishes. The granules have an affinity for acid dye. The nucleus is made up of two lobes connected by very fine threads. The granules play important role in immunity, allergy, and hypersensitivity. Basophils. The basophil stains with basic dye containing relatively large granules which are less abundant than in other types of granular leukocytes. The nuclei are twisted and show two or more constrictions. These corpuscles are seen infrequently in human blood, forming 0.5 to 3% of total leukocyte counting. The granules contain heparin, histamine, serotonin, hence these probably present the mast cells of connective tissue.